Human development starts with fertilization, which is the moment when a sperm and an oocyte, or egg, fuse to form a zygote, the seed of what will eventually grow into a human baby. During sex, semen containing 200 million spermatozoa, aka sperm, goes into the vagina. And that seminal fluid is alkaline, meaning it helps neutralize the acidic vaginal fluids. The sperm quickly make their way through the cervix and uterus and into the fallopian tube, also called the uterine tube. Eventually, they get to the ampulla of the uterine tube, and then the infundibulum, which flowers out right next to the ovaries. By this point, most of those 200 million sperm that entered the body during sex have died. They either get stuck in the vaginal mucus, lost in the cervix, or killed and absorbed by white blood cells. Only about 1,000 lucky survivors wait in the uterine tube for the egg to arrive. As those sperm wait, they start to rub up against the walls of the uterine tube, and that helps them remove the protective glycoprotein coat and plasma membrane covering the acrosome, which is a cap-like structure covering what you might think of as the sperm's head. And this process is called capacitation. Once those protective layers are gone, the sperm can secrete an enzyme called hyaluronidase, which can break down hyaluronic acid, a major component of the extracellular matrix protecting the egg. Now, the egg is the largest cell in the human body. It's big and round and about the size of a grain of sand, whereas sperm are the smallest cells in the human body, only about 1/30th the size of the egg, and these guys are long and thin. The sperm first makes its way past the corona radiata, which is a layer of follicular cells. It then makes its way to the zona pellucida, which is the layer of extracellular matrix of glycoproteins that protect the egg. Only about 500 sperm cells make it to this layer. The zona pellucida is also called the jelly coat, since it's a clear jelly that covers the egg. And this sits over a layer of proteins known as the zona pellucida sperm binding protein 3, or ZP3 proteins for short. As the sperm gets really close to the zona pellucida layer, an acrosomal reaction happens, which has two parts. First, the sperm releases acrosin, which is a hydrolytic enzyme that bores a hole in the jelly-like layer of the zona pellucida. After that, the sperm starts assembling actin proteins that fold out like a large protein crane that anchors and binds the ZP3 proteins. Once the sperm's anchored to the surface of the egg, the plasma membrane overlying the sperm and egg fuse, and this is called sperm binding. Now, inside the egg, there are cortical granules, which are like bags of enzymes like peroxidases. When a sperm binds to the egg, even though it's not really clear how, calcium levels start to rise in the egg cytosol. These rising levels of calcium signals the cortical reaction, which is when the granules fuse with the egg cell membrane. This releases enzymes into the space between the surface of the egg and the fertilization envelope. Those granule enzymes get mixed in with glycosaminoglycans, water, and calcium ions to form a gel-like barrier called the hyaline layer. This prevents other sperm from getting inside, and it ensures that only one sperm can fertilize the egg, and therefore prevents polyspermy. So that's how a single sperm out of the hundreds of millions successfully enter the egg. Just to put it in perspective, that's about the odds of winning a major lottery. While this is all happening, the egg undergoes its meiosis too, which leaves a second polar body which has barely any cytoplasm and usually undergoes apoptosis. The surviving daughter cell ultimately forms the mature female ovum, or egg. The nucleus of the egg is called the female pronucleus, and it has 23 chromosomes. Now that lottery-winning sperm sheds its sugary protein coat completely, leaving it behind on the egg's surface, and uses its tail and mitochondria to make one final short swim over to the female pronucleus, which is the nucleus of the egg, settling down beside it. Once it's at rest, the sperm's tail and mitochondria detach and degenerate, and the sperm's nucleus expands a little bit and becomes the male pronucleus. As the male and female pronuclei get closer together, a groove forms between them, and their individual nuclear envelopes dissolve. Those pronuclei release their genetic information, and a structure made of microtubules called the mitotic spindle forms across the diameter of the cell, and it weaves the chromosomes into complementary pairs, aligning them at the equator of the cell. At this point, the cell contains both maternal and paternal genetic information, and we've formed a zygote. This moment in embryonic development is called syngamy, where there's a single cell, but it only lasts a few hours before the process of cell division starts. Alright, as a quick recap, fertilization happens when a sperm successfully navigates through the vagina, cervix, uterus, and uterine tubes to meet the egg. From there, it makes its way through the corona radiata, the zona pellucida, and the oocyte cell membrane to fuse with the egg. The genetic materials contained within the nuclei of the sperm and egg combine to produce a brand new diploid cell called a zygote.